So good evening and welcome to the first of the 2015 Darwin College Lectures, which this year are on the broad theme of development. And welcome not just to everyone here in the Lady Mitchell Hall, but to all of you who are on the video link elsewhere on the Sidgwick site, and also to the many thousands of people around the world who watch these lectures later on. They are freely available on the web from sometime next week. So Darwin College is 50 years old, founded as the first college in the University of Cambridge that was exclusively for graduate students, and the first mixed college. From a small beginning, we are now amongst the largest colleges, with an alumni body encompassing every subject and spread right around the world. I think a real example of very successful academic development. The first lecturer in this, the 30th series of these weekly Darwin College lectures, is Sir John Gurdon. John Gurdon is a developmental biologist who has been called the godfather of cloning for his famous work on cloning frogs. That he would end up as a scientist was not particularly anticipated. His abilities were not recognized at school. Coming bottom in his year in science, his report said, "I believe he has ideas about becoming a scientist. On his present showing, this is quite ridiculous." Nevertheless, he went to Oxford to study classics, but changed immediately to zoology, continuing with a doctorate on frogs. After posts at Caltech and then back in Oxford, he moved to Cambridge to the MRC, Laboratory for Molecular Biology, the LMB, then the Department of Zoology, and then as a founder member and the first director of the Wellcome Trust Cancer Research Institute for Cell Biology and Cancer. It's not called that now. In 2004, it was renamed the Gurdon Institute in recognition of his many achievements. John has also served for 7 years as the Master of Magdalen College. At the same time, he was still active in the lab. He's a Fellow of the Royal Society and the Academy of Medical Sciences, and his awards include the 2012 Nobel Prize for Physiology or Medicine, made for the discovery that mature cells can be reprogrammed to a pluripotent stem cell state. So I very warmly welcome Sir John Gurdon to speak on reprogramming animal development.
So thank you very much, Professor Fowler, for your introduction, and also perhaps I could thank Torsten Kruder for having involved me in this. Great pleasure to be able to contribute to this series. So that is the subject of what I want to talk about. It's probably appropriate to talk about development because we all start as an egg. And it's a good question of how the egg decides to turn into what we are now. That's the animal side of development. And I'm then going to talk about a few other things. So I'll start talking about the normal development. So how do you go from an egg to an adult? I'll show a few pictures of what it actually looks like, and then discuss the question of how does an egg know what to do, no one there to guide it. Then the second part of the talk will be whether we can hope to go backwards, from the adult back to the egg again, and hence to embryo cells. And so can new cells be made from old cells? Can we replace old cells with new cells? And to the extent that we can, what are the ethical and legal constraints on making this procedure generally available? So starting then with normal development, how does an egg progress to being an organism or animal. And this is a video taken by Jim Smith, and it should show how this works in the frog egg, which is large and therefore easy to see. This is how development really starts. Once the egg has been fertilized, here are several of them, and you can see that they pretty much develop in time to each other. They start dividing into lots of little cells. The egg stays the same size. It's generally true of development. And this takes only a matter of a few hours after fertilization for the egg to form a ball of cells, some few thousand of those cells. When that's happened, the next stage is that the cells start moving inside this ball of cells and start forming structures. So here you see the cells moving inside to this ball. Very soon it starts to elongate, and that's the future brain, head, and within a couple of days, the egg has turned into the basis of an organism. It now has a head and many structures. So that's what development actually looks like in those cases where you can see it easily, as in the case of frogs and some other animals. So this process can be summarized by saying that life starts with the egg, forms an embryo, and then in mammals, the fetus, and in due course, the various tissues and organs appear. Brain, nerve, skin, hair, blood, muscle, heart, bone, and everything else. So the way it works is that cells start to, on a certain pathway, they make, at an early stage, they make the kind of cell which will form these structures. Other cells go in another direction. They form cells whose progeny will form these kind of different tissues. So the question is, how do these arise? And this is really the key question. How does a fertilized egg know what to do? Because you realize that in most animals, there's no one to tell it what to do. In fact, you're all familiar with frog spawn. This is what frog spawn looks like in a pond, all these eggs. And with no guidance, in a matter of a few days, each one has turned into a tadpole. And they sit there till they eventually escape. So it's a pretty amazing process as to how those eggs, with no guidance from anyone, somehow they know how to turn themselves into these more or less complete organisms. Now, there were a number of ideas. People have been intrigued with this idea for a long time, and you can see that one idea was that the sperm, which they couldn't see into very clearly, but they thought maybe the sperm had a little man inside it. You notice that the arms are neatly folded around the knees, so to pack this little homunculus, as they called it, into the sperm, they had this sort of idea. That would mean that really the major problem in development has already taken place, and the sperm already has it in it, and you just have to get bigger. That was the idea that existed for quite a long time. And there was a famous German embryologist called Dr. Haeckel from that date, and he was an immensely powerful person at that time, so much so that they felt that if he stated something, it must be true. And one of his statements was that phylogeny is the mechanical cause of ontogeny. Rather long words, difficult to know quite what they meant, but most people felt this did not elucidate the problem at all. And in fact, I think I would describe him as what I think we call the whipping boy. In other words, when something was inexplicable or clearly didn't make any sense, they all blamed it on Haeckel. And this is a common view, and I've now summarized one of the comments about Haeckel, and it said, you see, this is a kind of report, almost as bad as my own report, but what it said was that Haeckel's greatest disservice was not his total ignorance of exceptions to his rule, but his emphasis on his irrefutable explanation of the mechanical cause of development. That was fairly bad, but it went on to say that he thereby distracted those who might otherwise have made a valuable contribution to the whole field. So that's a fairly substantial report, at least it was posthumous. But so he was the one that people always blamed when they couldn't understand anything at all. And I can, in a way, sympathize with that view when you read his what he felt was important. So now Haeckel was Herr Doctor Professor, very important list of things, and of course they like to be known as that, but I want to draw a contrast between Haeckel and one of the most famous people in Cambridge, namely Fred Sanger, who doesn't call himself Mr. Doctor Professor or anything else, was just Fred Sanger. And he's, as you may know, probably the only person that I'm aware of who won 2 Nobel Prizes in the same field. Quite extraordinary. So if you were to, and he was extraordinarily modest to an amazing extent, but if you were to change his title from Fred Sanger to what it might actually have been, it would be Professor Sanger, FRS, Order of Merit, Companion of Honor, Commander of the British Empire, Nobel squared because he got 2, and PhD to the 27 because he actually had a PhD, but he had so many honorary degrees that he was about a doctor 27 times over. So he's one of the great people of this community, and if it weren't for him, we would not be knowing all that we do know about the genome, and even such remarkable things as being able to trace the lineage of, I think, King Richard III from many hundred years ago to the present depends ultimately on sequencing, which Fred Sanger initiated. As a matter of fact, my own ancestors go back to the year 1200, and I'm trying to negotiate to see if they could take one tooth out of the skeleton, which, as a matter of fact, was found recently when they had to relocate a car park. If you get permission to take the molar tooth out, then they can determine whether I have anything in common at all with this ancestor some 800 years ago. But I'm anxious to draw attention to Fred Sanger because he's a truly amazing person. Now, the idea that Haeckel had came about because he noticed that embryos of different kinds look somewhat similar. Lizard, chicken, deer, tarsier, superficially pretty much the same. So he thought that was a clue as to how things might work. And nowadays, if you look at the embryos of a mouse and a human, you can see that they're not actually that different, at least not different compared to a human and a mouse. And I actually forget for the moment which one is which. But that's how the same principle that early in life we look pretty much like anything else. Now, that says, going back to this idea of whether the egg or the sperm could have in it some kind of preformed human or body, this is actually a frog egg, and if you look inside, there is no indication whatever of any kind of person tucked up in there. It's just a mass of so-called cytoplasm, and this is all yolk, just spread. There's no indication of any kind of preformation at all. And even when the egg is fertilized, it looks much the same. It's the egg nucleus, male nucleus, so there's simply no apparent structure there at all. So how does the egg begin to know what to do? And there are 3 mechanisms. One is the asymmetric distribution of molecules, parental molecules. signaling between cells and a community effect. And I'll be talking about those, just say a few words about those different mechanisms. So talking first about the principle of the asymmetric distribution of substances in eggs, we can go back to work of a famous man called Conklin in 1905, and he made the observation that if you look very carefully at the egg of an organism called cellular, a mollusk, you can just see that the egg, the single egg cell, actually has some yellow-colored pigment at one point. So it's not as homogeneous as you might think. And when the egg divides, this yellow material divides too and gets distributed, and eventually the yellow material all ends up in cells which are going to be muscle. So the principle there is that the egg is already somewhat heterogeneous. It doesn't have heads and arms or anything else. It has substances in it which are asymmetrically arranged. And these substances get distributed as the egg develops. Similar thing in another organism, the cytoplasm at one edge is colored, and it all ends up in a lobe which ends up in one cell, and that cell with the lobe will become muscle. Now, the fundamental principle here is that you start with an egg or a cell, and there are some molecules shown by a black spot. As the cells develop, they make more of these molecules. At first, they are spread throughout the egg, and then when this cell or egg is about to divide into two, these molecules will go to one end of it, rather like the yellow cytoplasm of the mollusk egg. They go to one end, and then when this cell divides, most of these go into one daughter cell. And typically, that daughter cell is small, the components are concentrated, and this cell will then become something special. So in this case, it's skin, but the same would apply with brain cells. And then the remaining cell has a little bit of this left, and in course of time, it makes more and repeats the process. So the principle is that there are gene activation molecules. They accumulate an external signal which will move them to one side so that one of the daughter cells has a high concentration and the other one has a low, and these ones then are told to specialize into something, whether it's brain or skin, doesn't matter. That's the principle. Now, the second mechanism is signaling between cells. And this is a diagram to illustrate that. Here's an example of an egg, and it does have an asymmetric distribution of components. So the top end of the egg has a yellow component, more concentrated than the bottom end, with the blue components. So these cells are beginning to be directed towards the yellow fate, and these towards the other fate. Very soon, the blue cells send a signal, molecules to the yellow cells, and make some of them switch to something else, in fact, becoming muscle. So the yellow cells would make brain, the blue cells would make intestine, and the signaling causes some of the yellow cells to change fate and become muscle. That's another way of increasing the range of cells that are made. And this shows another remarkable fact, that the signal loses concentration as it goes from one end of the embryo to another, and cells get different amounts of this signal. It's called a concentration-dependent effect. Now, the third mechanism I'm going to mention is called the community effect. And the principle of it is this, that these cells release substances, little black spots, and if there are just two such cells, the concentration of the black spots is very low. If many of these cells are together, the concentration of black spots is very high and ends up creating a signal to the cells to make them change. Now, this community effect, curiously enough, was discovered entirely independently in a process called quorum sensing in a light organ. Same principle, but different system. This is a fish which lives deep down, and it has an organ there that emits light. and prey, which the fish is going to eat, come and attracted to the light when the fish then eats them. And this organ there emits light according to the community effect. And that's because it has a very high concentration of bacteria, and when there's a high enough, there is enough signaling between the bacteria with a particular molecule to make it emit light. So the idea that you make a concentration-dependent process also comes into this. That's the so-called community effect. Now, that's the first part of this talk, and it gives you just a feeling for how the process of development works, how go back to the main point, how a single egg cell, just one single cell, with no apparent structure in it, can gradually turn itself by these mechanisms into an organism that we recognize. That's the process of normal development, how we all started life, going forward from an egg to an individual. So the second question, which I'll now address, is can we make that go backwards? Can you go from an adult back to an embryo again? We have to remember that the process of development or differentiation is amazingly stable. We are used to the fact that in our brain we don't have skin cells, or in our intestine we don't have muscle. Happily so. So most of our tissues remain pretty homogeneous. And that's because once a cell decides to become something like that, all its daughter cells do the same thing. It's an amazingly stable process. However, it is possible experimentally to reverse that process, and that's what I'm going to talk about now. So in these experiments, which I'll mention, we have to go back some 50 years to remember that in the 1950s, nobody knew whether all the cells of the body had the same genes. So the brain and the skin and the muscle, for all we knew, might have different genes. Genes could be lost as the embryo develops. And this is now, we know as a result of these experiments that's not right. And this has led to the current concept of cell replacement. And the idea is this, that you start with something like skin or intestine, cells stably committed to these. Then you do this manipulation we call nuclear transfer, putting the nucleus of one of these into the egg, and the egg develops into the embryo. During this time, it begins to change, and then you can add chemicals to these cells, and you can direct them into something completely different, like nerve or muscle, having started as skin. So that opens up an enormous opportunity. If we could really take something like skin, which is easily available, and derive from it brain, nerve, muscle, or something else, these new cells would have the same genetic constitution as the original skin. And so if you imagine this happening in an individual, there would be no immunosuppression needed, because the new cells are genetically the same as the original ones. So it's an ideal way of going to the position of where you can do cell replacement in humans. And that's why this field is attracting such a large amount of interest at the present time. Now, this idea of cell replacement actually goes back a while, and there was a skull excavated dating from 1800 BC, a Mayan lower jaw. It has teeth in it, and these two teeth are false. The ones with a flat top, those are false teeth. That's not so surprising, because they might have felt that it would be good for the presumed afterlife that you should look in good shape, and so you put in some false teeth to present well. But the interesting thing is that these false teeth, you can determine if you're a dental expert, that the bone has reacted to those false teeth. So it means that the teeth were hammered in real life, and then the bone reacts. So that is probably the earliest example of replacement. Pretty uncomfortable, I would think, but they must have thought it was a good thing to do. Moving on, people have been attracted to this idea, and here you see a patient with needing a new leg. For some reason, this painting, which is from about 1495, shows a black person's leg being put in place of a white one. Rather nice genetic experiment. But and this is imaginary, but you notice that the nurses or assistants had wings. It was probably thought to give a better chance of the replacement working. But of course, in practice, this has not been a reproducible result, just an imaginary painting, and we have to go on from there. Now, it was this man, Weissman, who considered that the basis of development was the genes would be lost or inactivated. So as development was proceeded, he said maybe the skin cells lose genes for the brain and so on, and this needed this test that I mentioned in the 1950s. And the basis of this test is what we now call nuclear transfer, where you take skin, separate the cells, and inject one cell, actually it's a broken cell, into the egg. And in some of these cases, the egg will develop into a completely normal frog, or in the case of mammals, a mouse. And so the skin cell nucleus is able to, through this route, become reprogrammed and to generate completely new, all over again, a new complete animal, even though the nucleus of that cell would never normally have done anything other than remain a skin cell. So it's a rather dramatic change, and this procedure now works remarkably well with most mammals. I remember when I was put onto this as a graduate student, there were enormous problems. My supervisor said, well, you just inject a needle into an egg and put a nucleus into the egg. Well, it sounds rather simple, but in practice it was almost impossible, because if you took a needle and pushed it through this egg, the jelly surrounding the egg simply indented, and you couldn't get the needle into the egg at all. So it looked like a guaranteed failed experiment. But luckily, it turned out that if you ultraviolet irradiate the egg, that eliminates this kind of objection. So that was one hurdle, luckily overcome, only to find that there was another one, that it wasn't sufficient just to take a needle. You had to take a broken needle, which is very small. That's only about 10,000ths of a millimeter across, and you have to shape it into a shape like a sting of a wasp with a hole, but so sharp that it can go into the egg without damaging the egg. And that again required some effort to design the instrument with which you could shape the end of this micro needle into the shape of a needle, a sharp needle. But this eventually worked out, and fortunately, this worked well enough to get a normal adult frog. This was the first one obtained, 1958, and contrary to what many people say, this so-called cloned animal was completely normal. It lived for about 20 years and had about 5,000 offspring. So it was a perfectly good adult, achieved by taking the nucleus of a cell which had already started to become something, to go back again and start life again. And this, in course of time, led to the cloning, as they call it. When this clone was starting with an albino cell, injecting nuclei into eggs of the wild-type female, and all of the clonal progeny, of course, come out albino, and they are identical twins. And so people used to say, well, perhaps this is something we should worry about. How do you know you won't end up making clones of Hitlers or something like that? Even though I suppose we now know that if anyone had been able to make a clone of Hitler's, there's almost no possibility that they would have resembled in any behavioral context the way that Adolf Hitler did. But this was not really a scientific experiment, that was just a curiosity. And the important experiment was really to take something specialized, like the intestine of a tadpole. The green is the food of the tadpole, and you can do this experiment taking cells from this intestine, and by this procedure you can get these normal-looking tadpoles, this one from a fertilized egg, that one from an intestine nucleus, and when you look at the eye of these tadpoles, they're essentially normal. Lens, layers of retina, everything else. But remember that each of these cells, now constituting the eye, was previously the nucleus of an intestine cell. So it illustrates the transformation obtained by passing this nucleus through the egg. The egg has this rejuvenating activity. I comment briefly on Dolly the sheep, which was done in Wilmot in 1997, and it captured enormous interest, partly because he gave it a very good name, Dolly. And people said, well, why didn't you give a name to these frogs? The real answer was there were too many of them. We had about 500. So there wasn't, Dolly the sheep was almost unique. So we didn't give it a name, and that Dolly became very famous. Now, a derivative of this experiment was done by Wakayama, really quite interesting. He took a mouse and took a cell from the mouse, transplanted its nucleus to an egg, enucleated egg, and grew another mouse. And when he'd grown that mouse, he took the similar cell from this mouse, transplanted its nucleus to an egg, and grew another mouse. And he did this, amazingly, 25 times over. They're called serial nuclear transfer generations, some 500 cell divisions. And so this means that by starting with a cell like this cumulus cell, which would never have gone on living very long, you can give it an almost indefinite life. Almost a total rejuvenation going on in principle forever. So the lifespan of a cell is not to do with the cell. It can be changed by passing the cell continuously back through the egg, which has this rejuvenating activity. So the nucleus of this cell has an infinite lifespan, and I think particularly interesting is the fact that there was no cancer incidence in this case. Because normally we think that the longer you keep a body cell going, sooner or later, some of the daughter cells will get cancer. But if you can carry out this rejuvenation, that seems to eliminate that cancer. Interesting experiment. Now, I move on to the next important development. This was done by Martin Evans. Many of you may have known him. He worked in the genetics department in Cambridge, and he really created the idea of embryonic stem cells. And these are absolutely fundamental to this whole principle of cell replacement. And the reason is that if you can take embryo cells and keep them growing forever in culture in the laboratory, they don't grow old, they stay embryonic forever. And so you can take those at any time and take them as a resource and turn them into any kind of cell you want. These are the embryonic stem cells, and very appropriately he received his Nobel Prize for that in 2006. And this is, as I say, fundamental to the whole process of cell replacement. So I'm only going to now just to give you the conclusions from these embryonic stem cells. And I should mention that an embryonic stem cell is one that remains embryonic, as opposed to something like a skin stem cell, which will always remember that it's skin. The key thing here was to get an embryo cell to remain totipotent indefinitely. And so the conclusion from this is that cells can be grown from the early embryos of mice and indeed humans, and are known as embryonic stem cells. they can be grown indefinitely in the laboratory to make billions of unspecialized stem cells. They can be made at any time to differentiate or specialize into heart, brain, or other kinds of cells. So it was a complete revolution to think it's possible to keep a cell in this kind of permanently embryonic condition. Now, this is a video, I hope, which will show the combination of these methods that I've talked about. So let's see if this works. I think it's going to. Yes, there we are. Now, these are beating heart cells, cardiomyocytes, a whole lot of them, several thousand, beating together. And those have been derived from monkey skin. I'll just go back one step to show you how that works. So the experiment was this. You start with a monkey, transplant the skin cell nucleus to the egg, as I've mentioned, egg from another monkey, and then you do the cloning or nuclear transfer to make more cells from this egg. You then create embryonic stem cells from these cells by the Martin-Evans procedure, and you can increase this number as much as you want. You can then add chemicals to these cells and turn them into anything you want, including the beating heart. And that's the picture we've just seen of these beating heart cells. If I could get this back again. There they are. These are they're skin-derived cardiomyocytes. And that's kind of impressive. You'd think that might solve all the problems. It's not quite as simple as that, because you have to be able to transplant these cardiomyocytes, heart cells, into a patient in a way that they cooperate, they integrate into the patient's own heart. That at the present is very difficult to do, because these cells like to beat in their own way, irrespective of the way that the host heart is beating. So that causes considerable confusion. But that, I think, will be and it will eventually be solved. But it is impressive that you can make such splendid heart cells from the skin. So I will now very briefly run through one important thing, which is the mechanism by which the egg rejuvenates the nucleus. And in order to avoid boring people, I'm going to just skip a few slides to say that the way this has to be done is to inject nuclei into the nucleus of the growing egg cell, and you can get a highly efficient rejuvenation that way. And this video shows you how you can see the injected nuclei changing. They go from green to red because they've taken up components of the egg into these nuclei, which now resets the program. The two green ones are where you happen not to break the cell, and you have to break the cell to expose the nucleus to the egg components. But that now is beginning to tell us what the mechanism is, how it is that the egg rejuvenates these molecules. So we have a list of the various components, the molecules involved. This is only not complete at present, but the general principle here is this, that the egg is designed to transform sperm into an embryo-active nucleus. That's what the egg's meant to do. And it tries to do the same to transplanted nuclei. However, the nucleus of a specialized cell is designed to maintain the same pattern of gene expression, hence the stability of cell differentiation, and so it tries to resist change. So there's these two competing factors, and that's why the reprogramming is not completely efficient at the moment. So now, briefly, I'll talk about the induced pluripotency cells. This has been an amazing advance, dating from as recently as 2006, when Yamanaka and Takahashi announced that they could take cells of a specialized kind and can make them go back in development without having to use eggs. And the key importance here was that if you tried to do the reprogramming in the way I've mentioned, you would have to have human eggs. And that's very difficult to get. Understandably, women do not like to contribute eggs on any significant scale for this work, so you have to avoid the use of eggs. And this great procedure of Yamanaka and Takahashi achieves that. It's not always realized that the major pioneer of this work goes back to Weintraub in about 1980, when he discovered that there are particular gene products which have this extraordinary ability to switch cells from one fate into another. Sadly, he died of a brain tumor quite soon after this. But then Yamanaka came along much more recently and very appropriately got his Nobel Prize in 2012. And unlike the whipping boy that I've mentioned, he's a modest man. And when someone said to him, "How did you decide to do this experiment?" he was nice enough to say, "Well, my colleague Takahashi wanted to do this stupid experiment, and I had the good advice not to stop him doing that experiment." And the experiment amazingly worked so well. So it's been a real transformation. And what he did was to take skin from a mouse and transfer genes, these particular genes, into those cells. And amazingly, 1 out of a very large number, normally it was about 1 out of 10,000 cells, which have had these genes, was flip, shown by the red one, into the Martin Evans kind of embryonic stem cell. And so you select that cell, grow these cells, and get a pure population, switch them into nerve, which you can do, and then give it back to an animal. So that's the principle of induced pluripotency in adult cells. And these cells, they look like all other kinds of cells. You treat them this way, and eventually they expand, and eventually they form a colony of these switched cells. And when you grow these cells together, they eventually form complicated structures. And this bunch of cells, several 100,000, begin to form structures rather like a brain. And indeed, when you test them, they do indeed have similarities to the brain. And so the present direction of this work is to use this procedure, culture the cells for long enough, and keep them in a very compact form. When the cells start to signal to each other and create structures actually rather similar to brain. And here are some of the cells derived from the fibroblast body cells forming neurons, brain cells. And this is the main conclusion from this kind of work, from the pluripotent stem cells. These cells can create embryonic stem cells, and hence all other kinds of cells, from skin cells, without the use of eggs. And as a matter of fact, you can use these cells very conveniently to test drugs. So if you take cells from humans with neurodegenerative diseases, and you can grow them in the laboratory, you can test millions of different chemical compounds to find which ones can alleviate the condition. This is a direction that work is going in, particularly to try and relieve the human neurodegenerative disorders. This is not the only way forward, and people in Sweden for some time have been doing these experiments. They take fetal brain from aborted embryos, and you need about 3 or 4 fetal brains per patient. Of these, when you implant these cells into a patient, about 1/3 of them, the cells just disappear. In another 30%, the cells survive but don't do anything. In another 30%, the patient gets some benefit. So this looks encouraging from the point of view of Parkinson's and other disorders, but the real problem is that there's an enormous shortage of abortuses. Obviously, you can't get an infinite number. So that's a somewhat unrealistic prospect, but it is interesting that idea of taking embryo cells and implanting them into the brain can, at least in some cases, give some relief from these disorders. Now, this just summarizes much what I've been saying so far, that by the cloning procedure, you start with an adult, cells, like skin, and you transplant them either into eggs or oocytes, or you use the Yamanaka procedure. You convert them into embryonic stem cells and ultimately derive all these cell types. As a matter of fact, you can take some of these factors and switch cells between two sorts, like blood macrophage, blood lymphocyte, or pancreas exocrine to pancreas endocrine. Now, this procedure is useful, but it only works to a very limited extent. You can only switch between two kinds of blood or two kinds of pancreas. To go right back to the beginning again and derive everything else, you really have to get back to the embryonic stem cell state. So now let me, in the remaining time, just take a step forward and talk about the relevance of this kind of work from the point of view of patients. So people will always say, well, you talk about frog experiments, but what good is that going to do me? And they might say, likewise, you can do these experiments with embryonic stem cells, but does it actually help the patient? And so now I'm going to talk about this particular disorder called age-related macular degeneration. There are two sorts, the so-called dry sort and the wet sort. The dry sort is to some extent alleviatable if you use factors which prevent vascularization. So that's usable, but the dry sort is really incurable. And this is, many people, at least of my age group, suffer from this, and it's a pretty disastrous condition, because if you have it, this macular degeneration, you at first lose your central vision, can't see anything at all. Eventually, you lose peripheral vision and you become blind. So it's a pretty dreadful disorder and not uncommon. So to what extent can this be alleviated by this cell replacement therapy? The way it looks is like this. So here's the condition, age-related macular degeneration. And normally, you have the so-called rods and cones of the photoreceptor cells, which are very closely in contact with the so-called retinal pigmented epithelium at the back of the eye. Now, when you have this disorder, the retinal pigmented epithelium cells start to die, like there's one dead there. And when these cells die, they can no longer support the survival of the photoreceptor rods and cones. So these start deteriorating as well. However, using these techniques, particularly the Yamanaka technique, you can create these retinal pigmented epithelium cells in culture. You take skin cells or fibroblasts, and they rather easily become these kind of neural cells. So they can be created, and when you do this, you can get a sheet of these replacement epithelium cells. Here they are. Here are the cells, the nucleus and the cell, and you put this sheet of cells into the back of the eye so that they make contact with the rods and cones of the photoreceptors. You insert this, and of course the dying ones just disappear. They don't matter anymore. But you put in new ones, and these new cells are created as a sheet of cells. You grow them in the laboratory, put them on a little plastic sheet, and the plastic sheet looks a bit like this, quite small, you see, about 3 millimeters by 6 millimeters, and these are several thousand of these retinal pigmented epithelial cells which are needed to support the continuing life of the rods and cones. This is being done particularly by the laboratory of Peter Coffey in London. So this procedure is, in principle, almost perfect, because it's not so difficult to make these cells, and the actual surgery used to insert them is very mild. It's no more complicated than a cataract operation. You can, local anesthetic, put this little sheet of plastic in the right part of the eye, and it will match up with the photoreceptors, and the patient can go home that day. And it doesn't involve necessarily staying in hospital. It's rather economic, and it's proving remarkably successful. Now, some caution is mentioned by one of the pioneers of these brain implants, Ole Lindvall. He wrote a little article and said, it's all very well talking about these wonderful new techniques, but you have to be a bit careful, because there are hundreds of companies who will advertise and say, whatever you've got wrong with you, come to us and we will give you replacement cells, with no evidence at all that they will be successful, and they charge a big fee for this. And people will say, well, it's worth a try. But in fact, he calls it medical innovation versus stem cell tourism. And so there's a big need for some bodies to check these services and see if they really have any chance of working. An organization did that, called the International Society of Stem Cell Research, and they published a list of those companies who they considered could do this properly. But that had to be discontinued because the companies employed the services of a lawyer who said, if you publish this list, we will sue you, prosecute you for sums of money which you can't afford, regrettably. So they had to remove this valuable list of who could do these things and who couldn't. That's a problem, still exists, but I need to comment on this. Why, you might say, is it that this kind of cell therapy, replacement therapy, can work with the eye, but not so far with things like the brain? And the reason is this, that we as humans consist of 10 to the power 13 cells. That's 10 million cells. It's a huge number of cells. And if someone has a heart failure, the heart has about 10 to the power 11 cells, or nearly a million cells. And a damaged heart, you can lose some 10 billion cells. So to replace this, you need this huge number of cells. Now, the retinal pigmented epithelium cells needed for the eye is immensely less. It's only about 50,000 cells, which is considered a very small number. So that small number is realistic. You can make those safely, and they're very specialized cells, and they can then bring about this therapy, which at present is really not realistic for very large parts of the body. So that's why the eye is the chosen organ or tissue to work with at the present time. So now the question is, age-related macular degeneration, why can't we have this treatment now? Good question. And the fact is that surgical treatment needs approval by regulatory bodies. Regulatory bodies are extremely conservative. And you can bypass this by saying that you will guarantee not to sue the hospital or the doctor, so you can bypass the regulatory bodies in principle if you sign away your legal rights. But you have to prove that the person who is doing that has given what's called informed consent. And that's where the lawyers become, in my view, such a nightmare, because they will say, even if you've written that you forego this right and you are informed, they say, how do you know you're informed? So I asked a lawyer about that, and he said, oh, well, the way you do that is, if I wanted it, I would have to go to someone who the lawyers think is reputable, like a general practitioner, tell the practitioner what the procedure is, because he wouldn't know. He then has to explain it back to me, and then he has to sign that I have understood what he told me. It's a most intricate procedure. And even then, a lawyer is able to say, well, I'm not so sure that you really were informed. So this is a major blocking point. Now, I'm sorry, we've had these slides, but there are finally some ethical and legal issues concerning this, and I'll just finish by talking about those. So we have to say, what about the liability of the hospital, the surgeon? Should the National Health pay? And there's a good reason why they should, because the cost of a blind person is about $20,000 per year. The cost of this replacement is about $5,000, and it lasts indefinitely. So it would be economic for the National Health to provide this service for people suffering from impending blindness. Are you allowed to pay privately? Well, that's an ethical issue, but why not? And then finally, can the patient choose? That's the big issue, because we have this problem of lawyers who say, well, the patient can choose, but that doesn't really give safety to the hospital or the doctors. So that's a serious problem, and I think in the end, one has to say who is going to take the decision. You could say, what about the lawyers? Can they take a decision? Or the priests, or politicians, or even the patients? And I'm one of those who think that it should certainly be the patients. They should be, I think they can be informed, and they should be allowed to decide on their own risk. And as far as I know, everyone I know who is suffering from this macular degeneration and impending blindness would always say, yes, I'll certainly volunteer for this, because the worst possible outcome of such an operation would be some developing cancer. But if you're of an advanced age, that's usually not such a problem, and it would totally transform the life of so many people if this could take place. And in my view, I put the blame on lawyers, actually, and the reason they're the whipping boy. That's because lawyers in some way are able to give these punitive punishments, millions of dollars of compensation because something doesn't work. That is very discouraging to the whole medical profession if there can't be any safety, and if there is no safety in doing these things. So I think they're the ones that we should try to persuade that they should take a more reasonable view and allow patients to decide what they themselves would like to do. So that's really the review of this field. I've mentioned the costs, and here's the summary of what I've been saying. Fundamental is that all cells of the body have the same genes. If they didn't, this therapy would never work. Adult cells of one kind can be reprogrammed either through embryonic stem cells or directly to cells of an unrelated kind. You can turn skin into brain or eye cells. The replacement of some kinds of aged non-functional cells in human is, I would say, a real prospect in the near future if we can avoid the intervention of lawyers. Yeah, thank you.
Thank you very much indeed. I'm sure that the great majority of us here have childhood memories of a jam jar full of frog spawn that we've fished out of a local pond and then anxiously watched and waited as the tadpoles develop, and then they slowly grow legs and the tail shrinks, and ultimately there are a few perhaps little frogs to be released back to the wild. But all of that depends on the development we've had explained to us, but of course we have to keep feeding them with a bit of meat on a string dangled in that jam jar. That development is not necessarily a one-way process, that even the very boundaries of mortality can perhaps be reversed, is surely amongst the most astonishing discoveries of our age. And if you think to Gulliver's Travels, Gulliver on his travels met the Struldbrugs, immortal but aged and decrepit. The good thing was that at least they could be disinherited. So the question is really, can humanity face the even tougher challenge one day of old minds in young bodies? And must we love ourselves so much that we shrink from death? But these things are in part matters for another generation, and I guess probably their lawyers, though many people would hope it wasn't the lawyers as well. Here tonight, though, we must thank John for speaking to us and for his marvellous gift here to humanity. So thank you for taking us on a fascinating journey from tadpoles to frogs to stem cells and on to the prospect of really helpful medical therapies. Thank you very much.